The previous lecture covers how the action potential is generated and how it travels through a neuron. The neuromuscular junction is a specialized synapse containing a branch of an alpha motor nerve to one of the scattered muscle fiber within the total pool of muscle fiber included in the motor unit. This figure provides a scanning electron micrograph of an alpha motor nerve diverging to form neuromuscular junction at neighboring muscle fibers. M is muscle fiber, N is nerve, B is the nerve branch, cap is a capillary. For some muscle fibers have more than one neuromuscular junction. Interestingly, the divergence of the alpha motor nerve means that neighboring muscle fibers can be from different motor unit as well as from different motor unit type. However, all diverging nerve branches innervate the same type of a muscle fiber. When the nerve component of the neuromuscular junction is removed, the subneural folds are clearly seen as through the invagination along the sarcolemma. As previously explained, unlike the synapse, the postsynaptic membrane is not nerve but the sarcolemma of a skeletal muscle fiber. For all neuromuscular junction, the neurotransmitter is acetylcholine. There is some evidence from animal research that the neuromuscular junction is a response to exercise training by increasing the area of the neuromuscular junction. An illustration of the function of the neuromuscular junction is present in this slide. These are the ligand gate, also called a chemically gated sodium channel. These are the voltage gated calcium channel. These are synaptic vesicles containing neurotransmitters, which are acetylcholine. These are release neurotransmitters, which are acetylcholine. For the neuromuscular junction, the role of calcium uptake is the same for any synapses. However, the vesicle contain acetylcholine, and within the cleft of the neuromuscular junction, the enzyme acetylcholine esterases is present to rapidly convert acetylcholine into acetate and choline. The previous lecture covers how the action potential is generated and how it travels through a neuron. A motor nerve impulse action potential arrives at action terminal of the neuromuscular junction. There are voltage gate calcium channel, and these voltage gated calcium channels are activated by action potential. Calcium ion rush in and react with synaptic vessel. Synaptic vessel fuse with cell membrane. Acetylcholine released via process of exocytosis. Class, if you want to watch exocytosis and membrane fusion, I posted a YouTube video on this week module on Canvas site. And acetylcholine bind with the receptor site on a motor end plate, causing depolarization of motor end plate and sarcolemma, and impulse travel to T tubules and sarcoplasma reticulum. The ligand gate, also called a chemically gated sodium channel along the sarcolemma at the neuromuscular junction, are regulated by the binding of acetylcholine to the acetylcholine receptor. The sodium that then enters through the sodium channel of the sarcolemma raises the membrane potential to above threshold, trigger the voltage gate sodium channel, and causing the generation and propagation of the action potential across the sarcolemma and inward down the T-tubule network to the triad junction with the sarcoplasma reticulum. The value of resting membrane potential vary from cell to cell and range from about minus 20 millivoltage to negative 20 millivoltage to negative 100 millivoltage. For example, in a typical neuron, its value is negative 70 millivoltage. In a typical skeletal muscle cell, its value is around negative 90 millivolts. And a typical epithelial cell, its value is close to negative 50 millivolts. The threshold potential for most excitable cell is about 15 millivolts less negative than the resting membrane potential. In a nerve, if the membrane potential increases from negative 70 millivolts to negative 55 millivolts, the cell fire an action potential which propagates along the axon. In a muscle, if the membrane potential increases from negative 90 millivolts to negative 75 millivolts, the cell fire an action potential which propagates along the sarcolemma. Acetylcholinesterases is an enzyme that degrades acetylcholine, producing choline and an acetate group. It is mainly found at neuromuscular junction where its activity serves to terminate synaptic transmission. After a brief period, acetylcholine diffuses away from its receptor site and the ion channel closes. Acetylcholine is then broken down by the enzyme acetylcholinesterases. Acetylcholine is made from choline and acetyl coenzyme A. In the synaptic cleft, acetylcholine is readily broken down by the enzyme acetylcholinesterases. And choline and acetylcholine can be washed away from the neuromuscular junction or taken up by the presynaptic membrane of reincorporation back into acetylcholine. There is also some evidence that the neuromuscular junction may be a site of fatigue, muscular fatigue, during repeated intense muscle contraction, whereby inadequate resynthesis of acetylcholine prevent rapid, continuous bout of depolarization. Evidence also revealed the inability to rapid pump calcium ion pump into the sarcoplasmic reticulum in the recovery period between successive contraction, thereby preventing sustained neuromuscular junction stimulation of muscle contraction.
Today, Friday, marks the 20th anniversary of a nerve gas attack in the Tokyo subway by an apocalyptic cult known as the Armed Supreme Truth. 13 people died and more than 6,000 were injured. On Wednesday, for the first time ever, Japanese police authority released the audio recording of the communication between police when the attack happened. And we want to warn viewers that you may find some of the images disturbing.
日比谷線の八丁堀の駅、気持ちが悪くなった者2名がいるそうです。事故当やもしれませんが、詳細判然としません。
This is the first alert of the atrocity by the Supreme Truth cultists, and in the coming hour, calls for help and emergency reports would flood in as chaos overtook the Tokyo subway system.
突然、新聞紙に入った液体、これが落ち、この液体は当初どこにあったわけですか、どうぞ。満員の電車内に突然、
The liquid was sarin nerve gas. 5 cult members had brought it in on the trains during rush hour, puncturing the packages with sharpened umbrella tips. Sarin is so toxic that a single drop can kill a person. It was being released into the air as thousands of unsuspecting commuters got on and off the trains. And within an hour, 13 people were dead and more than 6,000 injured. But as investigators later discovered, it was just the beginning of a conspiracy.
The Armed Supreme Truth Court planned to produce tons of sarin and disperse it in downtown Tokyo. If they had succeeded, we cannot imagine its consequence.
13 armed members, including its leader Shoko Asahara, are on death row. The murder trial of the final suspect, Katsura Takahashi, began in January after his arrest in 2012. But public concerns over the sarin attack are far from over. 2 decades later, Japan remains largely baffled by why the cultists, including scientists and doctors who have graduated from the country's top universities, launched the attack. And the world is worrying that the nerve gas, which is so fatal and easily produced, could bring more harm in the hands of extremists. Nathan Rogers, CCTV.
Sarin is a highly toxic nerve agent produced for chemical warfare. Sarin is a potent inhibitor of the enzyme acetylcholine esterases. Since acetylcholine esterases is responsible for the breakdown of the neurotransmitter acetylcholine, the inactivation of this enzyme results in dramatic elevation of acetylcholine level. That's following with 1 to 10 minutes after direct inhalation due to suffocation from lung muscle paralysis. This is a reflex of proprioception. Knee-jerk reflex, also called patellar reflex, sudden kicking movement of the lower leg in response to a sharp tap on the patellar tendon, which lies just below the kneecap. In reaction, this muscle contract and the contraction tendon to straighten the leg in kicking motion. So in this case, quadricep muscle should be contracted. At the same time, hamstring muscle should be relaxed. How can body do this at the same time? Neuron can have both inhibitory and excitatory synapses. Neurotransmitter glutamate activate chemically gated sodium channel, excitatory positive synapse. So these channels are activated, it drives sodium along their concentration gradient toward the interior of the next neuron. So it can make afferent impulse action potential, cause contraction of the stretch muscle that resist or reverse the stretch. Neurotransmitter glycine activate chemically gated chloride channel inhibitory synapses. So these channels are activated, it drives chloride along their concentration gradient toward the interior of the next neuron. You should know this chloride is negative. The membrane potential to become more negative than the resting membrane potential, like a hyperpolarization, so inhibiting contraction of muscle. If you hit this patellar tendon using a hammer, the spindle sense stretch or speed of stretch and send afferent impulse to spinal cord. This axon release glutamate activates chemically gated sodium channel. So sodium rush in this cell body of a neuron. So afferent impulse action potential cause contraction of the stretch muscle, quadricep muscle here, that resist or reverse the stretch. This axon release glutamate activate chemically gated sodium channel. So sodium rush in this cell body of a neuron. So afferent impulse or action potential cause activate this interneuron. However, this interneuron release neurotransmitter glycine activate chemically gated chloride channel. So chloride, which is a negatively charged molecule, which make this cell body of neurons membrane potential more negative, inhibiting contraction of antagonist muscle, which is hamstring muscle. Does it make sense? Do not worry about this terminology. If you take KINS 425 or 456, you will learn this term. When Golgi tendon organs are stimulated, these receptors inhibit the contracting muscles and excite the antagonist muscle. I'm not really have experienced the exciting the antagonist muscle, but I have a lot of experience to inhibit the contracting muscle by Golgi tendon organ. Let's watch our previous KINS 379 exercise physiology students' performance.
You got it, you got it, you got it, you got it. No problem. Keep going, keep going.
One more.
You got it, you got it. Good, keep going, you got it. Here, you got it.
One more.
Keep going there. Come on, come on, come on, come on. Come on, come on, come on, come on. Yeah, you got it, you got it.
10. Oh, okay.
8, 9.
Oh.
Do you think he said no, or I think Golgi tendon organ said no. Does it make sense? It senses the tension, so it's inhibit contracting muscle. Sometimes like a professional bodybuilder used the strap, so it reduces the influence of Golgi tendon organ, allowing a more forceful muscle action. And practice quiz, Golgi tendon organs monitor the length of a muscle, whereas muscle spindles are sensitive to tension in the muscle tendon complex. True or false? You can find answers right. And when stimulated, Golgi tendon organ inhibit contracting agonist muscle and excite antagonist muscle. True or false? When performing a volleyball spike, the athlete's arm is rapidly stretched just before the spike, which of the structure detect and respond to that stretch by reflexively increasing muscle activity. Muscle hypertrophy, transient hypertrophy is the increase in muscle size that develops during and immediately following a single exercise bout. At the gym, many people said pumping, right? Pumping. Actually, that is transient hypertrophy. The result mainly from fluid accumulation in the interstitial and intracellular space from the blood plasma. So transient hypertrophy, as its name implies, lasts only for a short time. And chronic hypertrophy is the increase in muscle size after long-term resistance training. Change in both the size of muscle fiber called fiber hypertrophy and the number of muscle fiber called fiber hyperplasia. Muscle fiber can split in half with intense weight training, but this is based on animal study, not human case. And each half then increase to the size of the parent fiber. But conflicting study results may be due to a difference in the training load or mode, and satellite cell may also be involved in the generation of new skeletal muscle fiber. Did you remember satellite cell? I explained previously for hypertrophy, right? When I explained hypertrophy. And hyperplasia has been clearly shown to occur in animal model. Only a few studies suggest this occurs in human too. Satellite cell and migrate to the damaged region and fuse to the damaged myofiber or align and fuse to produce a new fiber, either of which lead to a generated or new myofiber. So hypertrophy and hyperplasia. Hypertrophy for a human and hyperplasia for animal. Fiber hypertrophy, this is for human. Net increase in muscle protein synthesis, possibly increasing the number of actin and myosin filament and increase the number of myofiber. So hypertrophy is facilitated by post-exercise nutrition, and testosterone plays a major role in promoting muscle growth. So post-exercise nutrition and testosterone, I'll prepare some special lecture for this. Early research suggests that the number of muscle fiber in each person's muscle was established by birth or shortly thereafter, and that this number remained fixed throughout life. Whole muscle hypertrophy could result only from individual muscle fiber hypertrophy. So this could be explained by size of actin and myosin and number of actin and myosin and more sarcoplasm. But however, no change in number of muscle fiber because we talk about hypertrophy. So it's very debatable. Hyperplasia, the longitudinal splitting, this is for only animal. With recent training, and which fiber is responsible for hypertrophy? Type 1 and type 2 fiber are increased. However, type 2 is responsible for hypertrophy. Does an increase in muscle size always mean an increase in strength? Not really, because weightlifter is stronger than bodybuilder, but bodybuilder is bigger than weightlifter, right? Because weightlifter, they have more contractile protein, like filament, but bodybuilder, they have less, but they have a more muscle size, yeah, like a sarcoplasm. Doesn't make sense. Yeah. So based on bodybuilders' case, right, not always increased muscle size always mean an increase in strength. An important component of the strength gain that results from recent training, especially in the early stage are neural adaptation. So motor unit recruitment, frequency of motor nerve firing rate, better synchronization of a motor unit during a particular movement, for example, squat exercise, and other neural factors like increased activation of synergistic muscle to assist force production are important to strength gain. However, long-term strength increase are largely the result of muscle fiber hypertrophy. I believe you know Hercules or Samson, right? At that time, they do not have any gym, right? But how can they have this kind of muscle? So I believe there are no gene, but there are some mutation. So myostatin is a growth factor that limit muscle tissue growth. So muscle tissue growth. Okay. So it means higher concentration of myostatin in the body may cause the individual to have less developed muscle. So if Hercules or Samson had mutation of myostatin, it's possible they can get muscle easily, they can be strong without exercise. So this is the example of a myostatin mutation. So this dog has myostatin mutation, so it don't look like Hulk, right? And cow, we call Belgian blue, right? It has myostatin mutation also, so it has a lot of muscle mass. Even fish, even mouse, so mighty mouse. And human, right? Did you remember who is this human? But actually not this photo is for Liam Orchestra, but you know, when we start our chapter, right, our textbook introduce Liam Orchestra, is right? So we can increase muscle mass if we have myostatin mutation, but this is really mutation. But we are already born. We cannot get this kind of mutation. But you know, we have a training program. So I'll prepare some special lecture after exam 2. So maybe at that time I'll talk more how to increase our strength and muscle mass, and even increase muscular endurance as well as power. Thank you very much.